I just want to introduce me and Shloka. So my name is Sirjan. I am a PhD student in neuroscience at the University of Wyoming, and I'm going to be hosting this event with Shloka. Together, we are a few of the virtual events officers here at WIN UK, and we have a very exciting plan ahead. Next slide. We have a very diverse set of audience joining us. You guys are from all around the globe. We have people from Cambridge, but also outside the UK. That's exciting. And now we're going to introduce you to WIN UK. So what is Women in Neuroscience UK? Women in Neuroscience UK is the very first organization in the UK that has been designed to specifically empower female neuroscientists. And it's a network that has been founded by Lizzie English, who's a PhD student at the University of Cambridge. And I'm thrilled to now give the stage to Lizzie, who can further explain what Women in UK is about.
Thanks so much, Sirjan, and yeah, thank you both, Sirjan and Shloka, for hosting and organising this event. It's so exciting to be back for this event because this is our first one of the academic year and on an awesome topic as well. So I hope you're all excited for it. As Sirjan mentioned, I founded Women in Neuroscience UK from scratch, and this was over a year ago now when I was starting my PhD, and I realised that there weren't many female role models in my space, and I was working in a very male-dominated lab group. And I looked online for networks like this, but I couldn't find anything quite like it that existed. So I decided that I wanted to do something about that, and I set it up myself. And since our launch event, which was about a year ago, actually, it was in November last year, we've grown so much. We have monthly virtual events during the academic year, so do look out for our upcoming ones as well. We have regular insightful blogs on our website. If you want to try out as a guest writer, we're always happy to have people submit their ideas for blogs. We have a very active social media presence across lots of different social media platforms. And we also have our monthly newsletters as well, which have had a recent design revamp, and they're a great way to kind of keep track of all the exciting updates that we have here at Women in Neuroscience UK. And something top secret that I've not mentioned before to the wider community is that we have some kind of secret in-person collaborations developing at the moment for early next year, and we'll be sharing more information about these soon. But I'm so excited for us to continue our existing activities that we have virtually so that we can ensure we have people like you attending from all around the world. It's amazing to see people sign up, not just from the UK, but from so many different countries. Do let us know in the chat if you're joining us from anywhere exciting. But yeah, as well as that, we want to supplement it with some in-person activities as well. We've been constantly getting requests from you guys in the feedback forms that you're interested in us developing some in-person aspects. So yeah, we're really excited to broaden and deepen our connections through this. So keep your eyes peeled if you're UK based or you're able to travel to the UK for some of our upcoming things next year. And yeah, as I said, this is our first event of the academic year, and I'm going to be passing back over to Shloka and Sirjan to host this one. But before I do that, just some reminders for you. We have a feedback form that will be available in the chat where you can fill in what you think about this event. This is super helpful for us in terms of planning our future ones and letting our speakers know if there are, particular points that the audience really appreciated about their talks. And social media, we'll be sharing some links later to the social media pages, and we really would love for you to keep in touch with us on there, and so you can engage with other people in our community as well. And yeah, if there are particular points during the session that you want to share on social media, be sure to tag us because we always love seeing your responses on there too. And yeah, I think that's everything I had to say. I'm looking forward to engaging with everyone in the Q&A sections after the talks later. And I'm excited to learn along with you from these amazing researchers in neurodiversity and also academics who have those neurodiverse conditions and learning how they navigate the space. So yeah, let's get started. Back to you, Sirjan and Shloka.
Just a quick slide change, and Resha, please. Thank you. Yep. Hi everyone, thanks for joining our Neurodiversity event today. If you haven't joined us before, a big welcome to the Win UK community. Just a quick note why we're doing this event. This talk is crucial as it addresses the often overlooked topics of neurodiversity, encompassing a spectrum of neurological differences. It promotes inclusivity, empowering adults navigating ADHD while advancing research and breaking stigmas. By shedding light on neurodiversity, we work toward equal opportunities, fostering a more compassionate and inclusive society. Understanding the significance of neurodiversity encourages acceptance and empathy, valuing diverse perspectives and contributions. This talk serves as a pivotal step in recognizing the importance of neurodiversity and its impact on the society.
Sorry, I was muted. So without further ado, let's welcome our speakers for today. First up, we have Dr. Ellie Dermot. Dr. Dermot is a professor of neuroscience at King's College London, and she heads the ADHD Research Lab. She'll be sharing her insights in a talk titled Destination Neurodiversity Research, Route from the Midbrain Head Outwards. So you don't want to miss it.
Next, we have Dr. Malika Chatterjee. She's an assistant professor at Amity Institute of Neuroscience and Neuropsychology. She's also a Fulbright Nehru Scholar at Brown University. She'll be talking to us about exploring neurodiversity, looking into behavioral modifications with sugar and in zebrafish.
Up next, we have Julia Dabrowska, who is a second-year DPhil student at the University of Oxford. Julia's talk is going to be titled Female PhD with ADHD, More Common Than You Think, and it's going to be a powerful journey through her experiences.
Last but not the least, let me introduce you to Sophia. She is an MD PhD student at Albert Einstein College of Medicine in New York. She's on a mission to share her experiences, including the strategies of managing ADHD as an adult. Thank you. It's my absolute honor to be introducing our first speaker of today, Dr. Dermot. Dr. Dermot is a professor of neuroscience at King's College London. She started a career as an electrophysiologist at Sheffield University and now heads up the ADHD Research Lab at King's, aiming to better understand ADHD and neurodiversity more generally. She's also a passionate educator and has been teaching in higher education for over 20 years. Outside of work, she runs and is owned by a rescue greyhound. Over to you, Dr. Dermot.
Thank you. So I think I now need to share my slides, which I'm hoping will work smoothly. Let's do it on the window share. Oops, I'm not sure if that's worked. Has that worked? Because I've now lost it. Yes, I think it has. Okay. Thank you. Yeah. So I'm going to kind of do a little bit of a whirlwind run through, because as was just said, I actually started out my kind of research career as a in vivo electrophysiologist, and I started looking at midbrain dopaminergic neurons, specifically with reference to addiction, so not really with ADHD at all. And then through some kind of odd route, I ended up thinking about ADHD in terms of midbrain collicular functioning. And I'll just say a little bit more about that. So I guess most people know what ADHD is. I'm not going to spend ages talking about what it is. But one of the things that really struck me was the prevalence of ADHD, and not just the amount of people that have it, but the impact of the condition. So, you know, when we think of children with ADHD, we might think of them doing the obvious things, so being hyperactive, not paying attention, but we don't necessarily recognize that those children will also have quite difficult times socially interacting and forming friendships with their peers, and then later on as adults forming relationships. Divorce rates, for example, are higher in people with ADHD. We know the condition has some genetic components, but it's not like there's a single gene, and there's various environmental risk factors. But what's interesting about ADHD is actually we've got pretty effective drug treatments for it, but we don't really know why those drugs work. And when you start to look at the circuitry involved in attention, for example, there's obviously all these sort of big hitter structures, things like the prefrontal cortex, which are thought to be critical in all kinds of important functions. But there's also other structures tucked away and in the circuit. So actually, I think the circle on this slide has moved slightly, but it's actually, well, it's covering the label. But if you look at the circuitry involved in attentional processing, there's actually a role for a structure that most people ignore, which is called the superior colliculus. And that was the role I started investigating in relation to sensory processing and dopamine. And then from there, looking at ADHD, realized that this structure could also be implicated in ADHD. And when you start to look a little bit outside, I also think of it as looking outside of the cortical box, because we do tend to fixate quite a lot on what the cortex is doing in lots of things, and sometimes there are other structures involved. So when you start to look at that, you start to think, well, okay, here's this structure called the superior colliculus feeding into the circuitry for attentional processing. And this structure has a very powerful influence over dopaminergic neurons. It's also receiving serotonergic and noradrenergic inputs, all of which have the potential to be affected by the drugs that we know treat ADHD quite effectively. So when I first started investigating ADHD, I took what I think is probably a classic neuroscience approach, and I worked in rodents. Some of that work was in healthy rodents, some of it was in an animal model of ADHD. I did local field potentials, multi-unit recordings, I did some anatomy, and of course for ADHD, I was also looking at drug treatment, particularly amphetamine, which is the main treatment for ADHD. And that allowed me to do some really interesting things. So I'm not going to talk in detail about the results, but what you can see on the screen here is the dendritic spines in the colliculus. Nobody had managed to visualize those before, and with the help of some outstanding anatomists, I was able to, or my PhD student at the time, was able to characterize dendritic spines in the colliculus, but also demonstrate that there weren't really obvious effects of amphetamine doses on dendritic spines, which suggests that the effectiveness of the amphetamine in ADHD was not due to changes in postsynaptic structure within the colliculus, at least. So we were able to start to unpick the mechanisms of action of this drug that's been in use for 50, 60 years, and nobody really knew how it worked. We then looked at the other side of the synapse, if you like, and we looked at a substance called synaptophysin, which is important in determining how much neurotransmitter is released, and so it's a presynaptic marker as opposed to the spines, which would be postsynaptic. And we found there were changes in presynaptic markers, suggesting that that could be a way in which amphetamine might be altering activity in the colliculus. So all of that was great. I got to learn about some new techniques and had various PhD students working on this project, and I got to work with some brilliant scientists. The downside of this is this was a rodent model, or even a healthy rodent being treated with an appropriate drug treatment regime for people. And all of that didn't really tell us very much. And it was sort of during that time that I started to think, well, you know, really, I probably need to actually start to do some research in people, not just rodents. As much as the rodent work was exciting and interesting, what did it really mean for treatment? Well, one way to kind of bridge the gap is to say, well, if an effective drug treatment for ADHD, or any condition for that matter, has an effect on this structure of the brain, is there anything else that we could use that would also affect this structure in the brain? And then you start to look at the NICE guidelines, and you realize that if the aim of a treatment were to alter the colliculus, for example, and that's obviously not all it would do, but that could do part of it, then one thing we could do is look at other things that alter the colliculus. And from there, we started to see things like exercise in the literature, eye movement training. Neither of those could be done effectively in rodents. Certainly, you can do things like wheel running and treadmills with rats, but you can't do a huge amount of diverse exercise as you can with people. And that led me on to my first human project in the form of ADHD. and that was to look at aerobic and non-aerobic exercise. So there was already research out there, but that research had some quite significant gaps in it. A lot of correlational studies, so you couldn't determine cause or effect, and a lot of studies where they've not actually measured the key outcomes for ADHD, so attention and impulsivity, for example. So we did a study looking at cycling and yoga. So this was an acute study, so people just came in, did some cognitive tests, did some cycling or yoga, did some more cognitive tests. And what that research showed us was that actually yoga and cycling reduce impulsivity, but for yoga, that only happens for those with ADHD. So that suggests that you could have some benefit to exercise, different types of exercise, if you have ADHD. That work was only published relatively recently, and we're still looking at follow-up work. So just as a kind of, I said it would be a bit of a whirlwind, but just to recap where I started in rodents. I then moved to trials with people with ADHD, but they were still, I would consider them quite restricted, because I was sort of saying, well, you can participate if you're this age group, if you don't have these comorbidities. It was quite a narrow group of individuals that could participate. And one thing that was important was that we were after people with, and I can't think of a sort of nice way to say this, but we were after people with pure ADHD. So not also dyslexia or autism or other conditions. For the exercise study, we did allow depression and anxiety, provided there was no medication. But for our other trials, we haven't allowed those either. So these people were sort of artificially simplified people. They were a very limited subset. And it also assumed that, to a degree, it was taking a medical model. We have to fix ADHD. And for some people, that is what they want. They want to see a reduction or control in their symptoms. And I think as scientists, we have to respect that. But of course, for many people, ADHD isn't seen as a disability or an impairment. It's seen as a difference. And that is where I then gradually moved into neurodiversity work. And I'm sure most of you here know the kind of difference between the medical model and the neurodiversity model. One is viewing something as a deficit that we might fix. The other is viewing something as a difference that we might try and better understand and remove societal or systemic barriers. So the disability doesn't come from the condition. The disability comes from the environment that doesn't acknowledge the condition. Now, I'm not going to say too much about what research we did, but rather how we did it, because I think that ties in better with the theme of this evening's talk. And we did it by working with neurodivergent individuals. So the main studies we're working on at the moment are looking at cognitive load or mental effort in online learning in neurodivergent students. So we started the whole process by getting a group of students who we paid to work with us to help us design the research. And they helped with the ethics application, they helped with the design. We published methodological papers from their help with them, so they are co-authors. When we got to lab-based studies, so we started with questionnaires and focus groups, but when we got to lab-based studies, they actually came into the lab and ran through everything and helped us create a guide to the lab experiments that is neurodiversity friendly. So things like considering, you know, if you are neurodivergent, there's a possibility you have sensory sensitivities. So we went through various processes with them to work out the lighting, to work out how to fit the EEG cap, the processes, all to make the whole process better for the participants. Now, we do have some preliminary results of this study, so we are starting to see differences between the cognitive load in neurodivergent and neurotypical individuals. We're still collecting all the data on it, but almost I feel like the findings for me are almost a bit of an aside. What's actually been the most valuable part from my perspective is working with the neurodivergent populations to help design and conduct this research. We're going to carry on doing that. We're going to carry on with our kind of a foot in both camps. So we are still continuing to work on treatments for ADHD. So seeing it slightly more from a medical model perspective. We're currently have put in a bid for a grant application to work with an organisation called Neurodiverse Sport. We're also testing eye movement training, And our latest study launching, which I'm very excited about, is to look at the relationship between ADHD and the menopause, because obviously that there is a possibility of overlapping treatments for those. And as well as that, we're continuing with the neurodiversity work. So we have a priority setting exercise, which sort of extends our research advisory board into a UK-wide advisory board, because we're saying any person in the UK who's involved in higher education, neurodivergent or neurotypical, but obviously we're very keen to hear from neurodivergent students, tell us what you want us to research. So we're trying to involve people right from the start. And so I think I've kind of come on a journey from thinking, well, I'll start working with some rodents in a lab, to actually almost crowdsourcing the research from the population of interest. So that's a whistle stop of what we're doing. I just should point out, of course, that none of this work was done by myself. So right from where I started to right where I am now, lots and lots of people involved in it, doing the work with me. And that was all I wanted to go through. So I'll stop, see if I can stop sharing now, if I can work out how to do that. I think that's worked. Did that work? Have I stopped sharing?
Yes, that worked well.
Great.
Thank you so much for the wonderful talk, Dr. Damit. It was really insightful, and I think we definitely learned a few different things about the circuitry especially. So if in case anybody in the audience has questions, we are happy to take it at the end of the talk. So make sure you leave the questions in the chat. Anvisha, could we have the slide deck up again? Thank you so much. So swiftly moving ahead, our second speaker for today is Dr. Mallika Chatterjee. Dr. Chatterjee is an assistant professor at Amity Institute of Neuroscience and Neuropsychology in India, and she's also a Fulbright Neuro Scholar at Brown University in the US. For her research, she focuses on identifying the molecular mechanisms underlying various neurodevelopmental disorders, including autism spectrum disorder, as well as schizophrenia. Dr. Chatterjee, the stage is yours.
Yeah, thanks, Sajjan. Thank you, Women in Neuro, to invite me. It's a wonderful opportunity and actually interesting. It will be my first talk regarding neurodiversity, which is a fantastic topic and should be discussed upon, and also on zebrafish, which is my new pet peeve, you know. So I'm just going to start sharing my slide. Just give me a minute. I'm not seeing this here. Just give me a minute. Okay. I'm not seeing my presentation on the system. Oops. One thing, why is it not coming on this? Sorry. Something. Yep, no, all right, sorry. Can you see my slide?
Yes, we can see the slide deck.
Yeah. Hi everyone. Great to see all of you here and the interesting, diverse audience. Today I will share a little bit of my journey as a scientist and also the wonderful fun things that I'm learning here at Brown. Well, the theme is neurodiversity and inclusion. So each of us are unique in our behavior. And in spite of all of us having a generally common neural circuitry, and like humans, zebrafish also have basic neural circuitry, but each of the fish that we look into, each has its own unique personality. which is shown in the graph here. What we did here was to measure 3 behavioral parameters of the fish. That's excitability. That's how excited it is when exposed to a stimulus. Habituation is how you get used to a stimulus, and startle response in response to a sudden acoustic burst of sound or light, So these represent the 9 larvae at 5-day-old larvae. And as you can see, we have just measured the percentage activity, and each larvae has a different level of response. If you follow the purple versus blue and the red, each red line is zigzag. That is the first larva as compared to the second, as compared to the third, and so on and so forth, have a different number or activity. And that shows that each is unique in its behavioral parameters. In overall, when we do zebrafish study, we actually do a sampling, and therefore we study the trend or the behavioral trend, which can be summarized as supposed to be the behavior of the whole population. But on its own, each is unique to itself. Coming back to my journey as a scientist, I began in Presidency University, Calcutta, where I did my undergrad in human physiology. Then I moved on to Science College at Calcutta University, where I did, in fact, my master's in genetics. Then my PhD was from University of Connecticut Health Center, where I worked on 2 of my favorite models, mice and chick. Then I went as a postdoctoral fellow to Tata Institute of Fundamental Research back in India, Mumbai, where I worked mostly on mice. And finally, I landed up in a faculty position in Amity University, Noida, where I had started a zebrafish lab. And that's what I'm also learning more about in Brown. During my PhD tenure, I was looking at a very specific part of the brain called the thalamus. You see a sagittal section of the mouse brain here, and this green area is the thalamus, which sends out filtered sensory information from the periphery to the cortex. And I was basically understanding the question or answering the question as to what genes are responsible for directing the thalamocortical projections. That is, what genes determine which direction the thalamic axons will go. And what we saw was a transcription factor called GBX2 guides the thalamic axons correctly. If GBX2 is not present, various downstream transcription factors like LHX2, LHX9, etc. are affected in their expression levels, which subsequently affects the expression of receptors which are present on the neurons and which sense the molecules, signaling molecules in the environment. Respectively, ROBO1 and ROBO2 are affected. So that was the summary of my PhD work. But what was further interesting, and which was like a sort of carryover story, which I'm still trying to develop, is there was an additional molecule or a sugar called heparin sulfate proteoglycan, in abbreviation HSPG, which actually helps to potentiate the activity of these receptors binding to their ligands by binding to them. So HSPG is a type of modified sugar, and therefore our hypothesis was, is there a third-party role of sugars in thalamocortical axon guidance? What is HSPG? As I said, it is a sugar. So it's basically a polymer of sugar residues, which are extensively modified. One of the major modifications that I was interested in looking into was orthosulfation, that is sulfation at the ortho positions of these sugars, either at the 2-ortho or the 6-ortho position. Now, these long chains are found either on the surface of the cell, or they are also found in the extracellular matrix freely floating around. They have a lot of diverse functions, which is shown here in the schematic. Either they help in conformational activation by binding to molecules like anticoagulating agents. They help in concentrating various ligands like the fibroblast growth factors on the surface of the cell. They also help in enhancing protein-protein interactions, and they help in concentrating a lot of these ligands. So heparin sulfate molecules are a new area of interest in the field of neurobiology, wherein the concept of heteronome has been proposed, wherein different differently modified heparin sulfates determine the identity of different types of cells. And a cell to site in this example is interneurons, where it has been shown that different types of interneurons have different compositions of modified heparin sulfates. And how is it created? It is created in the ER after translation of the core protein. The heparin, the carbohydrate molecules are attached to it. And there are various enzymes which help in this reaction. The most important or one of the key fundamental enzymes which are involved are orthosulfotransferases, which help in addition of this 2-ortho or the 6-ortho sulfation group. Now, why am I interested in heparin sulfate? Because there is a link to various neurodevelopmental disorders, including autism, that shows there are various papers which shows that when you do not have heparin sulfate, or in fact, when heparin sulfate is not correctly formed, then you can have various types of socio-communicative disorders. And one of the hypotheses is that it also involves correct formation of dendritic spine via proper heparin sulfate interaction with the synaptic and spine molecules. So in order to pursue the question of the role of heparin sulfate proteoglycan, I chose zebrafish as a model system back in our lab at Amity University because it is a viable model where it is cost effective. You can easily set up high throughput screening strategies, which I'll share in a bit. And also it is easier to maintain. So therefore, I got up, I diversified, and moving on from mouse, I started setting up in February 2022 a small zebrafish facility at Amity. And here, as you will see, we have gone into establishing various neurobehavioral paradigms. We started with adults, where we were looking at the learning and memory, for example. Here you see the T-maze, which looks at conditioned learning paradigm, where we train our fishes and the fishes know whether punished, it doesn't go into the red arm. When not punished and rewarded, it goes into the green arm. And then we test for learning and memory under various conditions, like in an ASD model or in models of sleep deprivation, which is another area of my research, etc. This shows the tracking of the fish, which we do with a software called AnyMaze. And this is the real adult fish put in a jar for tracking. We also do neurodevelopmental behavior, and these are the pretty eggs that you see, which we obtain after breeding the adult fish. This is a 2-day-old larvae, and you can see the heart beating, which is quite exciting and beautiful to see. And talented students in our lab, in fact, undergraduate students and interns who have come to Amity, they have set up with me this learning and memory paradigm, wherein they have observed that there is we were efficiently training the fishes, wherein they learned to go into the green chamber, which was the reward chamber, as compared to the red chamber, which was the punishment chamber. And wherein then if you are sleep deprived, they do not go into any of the chambers, which is represented by the various types of sleep deprived fishes, which I show here. So this is just an example of what type of work these students are doing there. Students have also looked into the novel tank test, done the novel tank test with adults, where talented student Arkadeep, our master's student who just passed out, he used adult fishes and treated them with valproic acid, which is a known model for autism spectrum disorder, and seen where do the fishes settle in when put into this glass jar, whether they explore the top compartment and stay there more, which is considered to be more exploratory and less anxious, or they settle in the bottom compartment. And what we have seen in the model of where we administer valproic acid, they tend to avoid the upper zone more, which is not surprising because they're considered to be less exploratory in such a model system. He has also done the predator avoidance assay, wherein we put in a big fish, the Oscar fish, predator fish here, and then we put the small zebra fish on the other side of the compartment, and we put in various barriers and see where the small fish goes, whether it goes towards the Oscar fish, that is the near zone, or whether it occupies the farther zone, that is, it is afraid of the Oscar fish. Arkadeep has also done tracking, which is with in small larvae, which is 7-day-old larvae. And what he has shown that he has successfully tracked the animals when treated with valproic acid, and when treated with various concentrations of valproic acid, example 25 micromolar and 48 micromolar, he has seen that the larvae seem to stick to the side of the walls, which is a sign of anxiety, also called wall-hugging behavior or thigmotaxis. So when I came to Brown, I wanted to use small molecules and see which would change the sulfation pattern of heparin. I was talking about heparin sulfase, coming back to that. And I wanted to see what was the effect of using the small molecules on larval behavior. Robert Creaton's lab in Brown, the lab who's my host, actually looks at high throughput neurobehavior imaging. And there he uses a system wherein he uses 96 well plastic plates, and each well has a zebrafish, which is like a 5-day-old zebrafish, and they track it with a DSLR camera and for 3 hours. And each image that is captured, you see this dish here, each of them contains a zebrafish larvae, and each image for 3 hours, 1,800 images are captured for each well, and then various activity parameters are assessed using either Fiji, which is a freely available software, or using the Python, which is programmed into utilizing the Oscar, which is a high computing facility at Brown, and then we generate data for 384 larvae at a time. So the system is a high throughput and highly cost-effective system. And this is the kind of how the picture looks like when the camera takes a picture. Post-acquisition data analysis happens. And this 3-hour period, that is 180 minutes, is divided into 10-minute periods each, 1 to 18. And this is the first period of where acclimatization happens. Nothing is given to the larvae, no stimuli is given. For the next 7 to 14th period, light stimuli, moving light stimuli is given in order to evaluate OMR response, which is a very common response, typical response of fish larvae. And then from 16 to 18 period, the larvae is exposed to an acoustic stimuli. So first no stimuli, then light, moving light lines, and the last is acoustic response. Okay. And what we see, this is an example type of data sheet which we generate. That is to say, these are the 25 behavioral parameters. Each column represents a behavioral parameter of the zebrafish, and the rows represent each of the molecules that I have treated with. One includes trichloroethyl sulfamate, which inhibits desulfation of HSPG. Sodium chlorate at various concentrations causes desulfation of HSPGs, which is opposite to trichloroethyl sulfamate, and rotlerin also desulfates, but specifically at the 2 ortho positions. And what is meant by green and red here is red means EW is egg water, which is, and the MSO are controls. We have compared the data respective to the controls, and red here means there is more activity, and green here means there is less activity as compared to the MSO and EW controls. This type of data is generated, and each larvae is at least, the numbers are big, which at least like we can do 100 and more larvae, and in total each well we can image 384 larvae in a single experiment. And here we do a sort of clustering to understand how the molecules behave. So as you see, TCS, which is causing inhibits desulfation, expectedly occupies one side of the control data, whereas the other molecules are clustered together on the other side in terms of their behavior. And this is an excessive, accomplished by using cluster 3 program. And this is a successful behavioral profiling of developing zebrafish and analyzing 25 behavioral parameters. We can also further zoom into, like we don't only look into the forest, we look into each individual larvae, and we can also look into their average activity over the last 4 hours, which was interesting because in patients with ADHD, ASD, etc., startle response, how they respond to acoustic stimuli is something very significant and something we should look into. And therefore, here, as you see, the sodium chlorate seems to have a significant effect, more than that of the other molecule, that is TCS or the control DMSO alone. So this is just the kind of data that we are able to analyze using the high throughput system here. And finally, a beauty of the system is that once you generate the high throughput data, you can compare it with existing commercial libraries of drugs. So for example, there is a FDA drug library, which has already been published by the lab, which I took my data of my molecules and compared the behavioral parameters with my molecules with that of the FDA library molecules. And what we found was many of these molecules had interesting correlative or similar activity. For example, TCS had similar activity to active D2 receptor antagonists, or here, as you see, rotlerin, my molecule, had a similar activity to D2 receptor antagonist. This suggests 2 things. Either you could use these drugs, which are already FDA approved, to target some of the disorders that my molecules might be causing, or these molecules are acting in a similar pathway as the dopamine receptor pathway. And I'm reminded of Professor Domet's, the first Professor Eleanor's talk, wherein she was talking about the dopamine pathway. And I'm very interested to see how dopamine and autism spectrum disorder and ADHD pathways, neurocircuitry actually interacts. Okay, and in particular context of the molecules that I have been using to change heparin sulfate molecules. And so this is the beauty of the system that I'm learning, and this is very preliminary results which we are still analyzing right now, and we hope to know more in the coming months. And so this is just fresh off the oven. And this is the lab, and this is Robert, who's my wonderful host, and these are the wonderfully talented postdoc and research assistant Sally and Thais, who have been helping me with the data processing. And thank you for listening.
Thank you, Dr. Chatterjee. I'm sure we all enjoyed hearing about your career journey, and especially the tutorial on how to train your zebrafish.
Thank you.
Unfortunately, due to time constraints, we will not be taking a break. Do feel to get yourself a tea or a coffee if needed. This event will be up on our YouTube channel soon. Over to our third speaker for today, Julia. Julia is a second-year DPhil student at the University of Oxford. Her PhD focuses on combining behavioral, electrophysiological, and machine learning methods to better understand brain network activity and mechanisms underlying attention control in context of new learning in a diverse population. Take it away, Julia.
Hi, okay, so I guess it's my turn to hopefully slide share efficiently. If you just give me a second. Hopefully, oh, sorry. Can you see the presentation now?
Yep, it's perfect.
Okay. Yeah, so thanks obviously to our previous speakers. My talk is going to be less on the science, more kind of on the personal lived experience side. So I'm also one of the WIN members. I'm one of the research blog editors. And my talk is about basically having ADHD while being female, while doing a PhD. So first of all, when you think of ADHD, what do you think of? Probably think of running around, fidgeting, unable to sit still, having crazy ideas, taking risks, blurting out whatever's on your mind, and most importantly, primarily affecting school-age boys that grow out of it when they're older. If so, you're not alone, because quite honestly, that is the majority of the world's perception, if they even believe it exists at all. Sadly, it's also the perception of many teachers, parents, and doctors who could have diagnosed earlier if they were more informed about the range of potential symptoms. Of course, ADHD does exist, and it's not just an invention of today's society or lack of attention fueled by TikTok's technology and too much sugar. It's, as we've all heard and know, it's a neurodevelopmental disorder. It alters the brain's typical development, and approximately 7% of the world's adults are diagnosed with the condition. We've already had Dr. Domit's presentation on this, but the two primary components are hyperactivity or impulsivity and inattention, with most individuals leaning towards one, the other, or a combined presentation. And depending on which symptoms an individual has, the disorder may be more or less visible, but this doesn't make it any easier. If anything, inattentive or internalized symptoms of hyperactivity make life more difficult, but we'll come back to that later. Resultantly, many adults get very late diagnoses or go undiagnosed completely, especially those who are successful in academics. And this is even worse for women who not only tend to show symptoms of inattention rather than hyperactivity, and so are less noticed by their parents or teachers. Of course, men can have these symptoms too, but they also internalize and mask many of their symptoms due to social cultural norms. And we'll get back to that later. So I just wanted to touch base on some of the symptoms and struggles that we deal with, some of which are less known than others. So of course, we have the basic hyperactive ones: inability to focus, restlessness, excessive talking, interrupting others, acting without thinking, being forgetful, distractible, making careless mistakes, struggling with organization, and sticking to time-consuming tasks. We all know those. They're all important, common, part of the official diagnostic criteria, but we also have so much more than that. You have time blindness, executive dysfunction, emotional mood swings, cognitive sluggishness, getting overwhelmed easily with seemingly normal workloads or chores, rejection-sensitive dysphoria, decision-making difficulties, poor working memory, justice sensitivity, hobby hopping, task paralysis, overstimulation, hypersensitivity to taste, sound, smell, touch, oversharing, fatigue. The list goes on and on. Of course, different people experience different symptoms, so not everyone with ADHD will have all of these, and people without ADHD may also experience some of these too. But it's just so much more than the basic criteria that the NHS tells us. Most importantly, I think the naming of the disorder itself is very inaccurate. Rather than a deficit of attention, it's a deficit of regulation. Regulation of attention, yes, but also regulation of our impulses, motivation, emotions, general body state. So if you remember the question from the beginning, if you ask me for one word I'd just think of to describe ADHD, one word to encapsulate the whole experience, I think it would be intense. So with ADHD, everything is always too much or too little. Finding balance is basically impossible. So you either can't focus at all, or you can't stop working for hours until you finish that one task. You're either euphoric because the sun is out and the world is pretty, or you're lying in bed for hours, unable to lift a finger because someone you thought liked you didn't invite you to their dinner. Signing up for new societies, commitments, starting new hobbies, or being too overwhelmed to send an email, see a friend, and do your laundry in one day. Binge eating or forgetting to eat. Wanting to party, socialize, and see the world, or being too drained to come out of your bed to go get tea from the kitchen, just in case you have to speak to your flatmates. Not caring about anything or anyone, or vicariously living through your friends' problems and having a meltdown when you think someone criticized you, even if they actually didn't. So I've given too many now, but you get the idea. Life really is a roller coaster, and figuring out where you'll be the next day, hour, or even minute is extremely difficult and prone to change. So now we've kind of covered in ADHD, lots of the internalized symptoms, and obviously they can be present in anyone, but they're typically more frequent in women. So differing symptomatology is only actually one side of the coin when it comes to decreased diagnosis rates. So think about a man who's messy, forgetful, impulsive. His house is a mess, dirty dishes, piles of laundry lying around. He forgot to buy groceries on the way home from work, so now has to order in to feed his kids dinner. Last night went out with his friends, had a bit too much to drink, got a stupid haircut. The next day, his colleagues laughed at him, got angry, slammed the door in their faces. But hey, that's just guys, right? They're messy. Getting a spontaneous haircut is just brave and funny, and they sometimes get angry for no reason. And ordering takeaway for his kids just makes him cool. Now think about women in the same situation. What society is having a messy and disorganized house, no home-cooked meal for your children, chaotically irresponsible decisions, and emotional outbursts seemingly acceptable female behavior? None. I didn't think so. So because of society's view on what women should and shouldn't be, females are much more likely to mask or hide their ADHD symptoms, often by developing coping strategies, suppressing their impulses, or mirroring behaviors of others around them. You may think that makes it easier. After all, they do appear to be struggling less. But the mental load is not any easier at all. In fact, masking itself takes up even more mental energy. But because it seems like they can get by, nothing gets noticed. And even if girls seem slightly daydreamy, forgetful, or emotional, isn't that just what women are like anyway? Meanwhile, under the hood, the internalized hyperactivity and overwhelm leads to spiraling struggles, guilt, and poor mental health. Structure that helps them get through their daily lives breaks down as soon as they're left alone, leading to intense overwhelm and burnout. This is particularly common when undergoing major life transitions. In particular, many women only get diagnosed when transitioning to working life, having children, or like me, research-focused degrees that lack the structure that school and taught degrees have provided previously. So a little bit about me. I'm Julia. I'm a second-year PhD student in neuroscience at Oxford, and I got diagnosed with ADHD last year, age 23. Thinking back, it all makes sense. I've always been talkative. My teachers kicking me out of the classroom for disrupting others, getting really low effort grades despite really high academic grades. I wander off on countless tangents when attempting to tell a story. I can never sit through a conversation about fiddling, to my friends' amusement, usually by destruction, ripping up receipts, or my favorite, tearing apart the peel of a tangerine I just ate into tiny shreds. I experience extreme mood swings within hours or minutes. A sunny day can make me euphoric. Friends not inviting me to join their impromptu dinner can disrupt an entire afternoon. Painfully, I often also come across as rude, interrupting people, struggling to focus on what they have to say, despite really caring about them. I often speak before I think, and I regret my words immediately afterwards. I'm always late, annoying my friends for wasting their time, and rightfully so. But while frustrating, these don't necessarily signal a disorder. Some people are just more restless or talkative or emotional than others. So why would I or my parents, who come from non-medical backgrounds, assume it was anything but just my personality? Everyone's different. But the major problems arose when my academic performance was affected. So I always procrastinated my assignments and left everything to the very last moment while being a very extreme perfectionist, but it worked. However, when the material got harder in my final year of school, I realized I actually needed to put in some work, something that I was not used to and I was not capable of. While my peers revised for 8 hours daily before the A-level or the IB exams, I could do 4 on my best days, and I was unable to sit without a 2-hour exam without getting bored and distracted in the middle. And unsurprisingly, the effect showed. I underperformed and I missed my undergraduate offer as a result. I still did well compared to others in my year, and so rather than suspecting an issue, everyone, including myself, thought I'd just been too lazy to prepare, resulting in guilt and regret lasting the next 5+ years. Undergrad was similar. I got good grades, but rarely excellent ones. I struggled to finish coursework earlier than 4 a.m. the day before the deadline. I spent 3 hours running out of time in exams. My peers easily finished in 2. I got lovely comments like this one on verbal presentations. I'd never considered I might have ADHD until my 2nd and 3rd year, until one of my friends was just sending me too many reels that seemed relatable. On which note, I have no shame in admitting that social media played a part in discovering my condition, my journey. But unfortunately, the pandemic made it virtually impossible to get assessed, and the lengthy process really put me off. But then things changed. So I started my master's degrees, much more research than teaching, requiring lots of increased amounts of independent study and organization, which was very not helped by the COVID-induced complete lack of schedule of sport, social, and other events I usually busied myself with. So underperforming, yes, again, and a particularly horrible exam experience got the best of me, so I set to it. And surprise, surprise, I got assessed and I had ADHD, exhibiting signs of every single one of the official symptoms at some point in my life. So while I had been expecting at that point, the diagnosis still brought about a range of very intense emotion. Anger for not taking myself through the process earlier, for not being bothered to do it earlier, enabling extra time in exams and fulfilling my potential that so many of my teachers have told me I have. Frustration that so many of my struggles could have been ameliorated much earlier had psychiatrists 15 years ago been more aware of its prevalence in academically well-performing girls. Confusion, realizing that a lot of my traits are due to ADHD, leading to questioning my entire personality. To this day, I struggle a lot with reshaping my identity. I think about this at least once a day. But nonetheless, there's also a bright side. Perhaps most importantly, I feel vindicated. Many of the problems I'd been blaming myself for over the years were not due to my laziness, rudeness, or general disorganization, but could be directly explained by disrupted brain connectivity and neurochemical signaling. I feel more secure, confident, and accepting of my flaws, because while ADHD can't be used as an excuse for everything, it certainly plays a part. Around people I'm comfortable with, I mask a lot less, freeing up mental energy to do other things in my life. And many of the resulting traits I have are positive. I'm extremely keen to try new things, challenge others' opinions, emotionally invest into the well-being of those I love. I'm bubbly, brave, and never boring to be around, or so I'm told. So in terms of handling my PhD, I'm still figuring it out. I have a very flexible working arrangement as I don't do much experimental work, which is both a blessing and a curse. I know that I can focus much better when I do regular exercise, particularly running, so I prioritize it over many activities. I work better when I'm around others, so I try to go into the office if I can. And I try to schedule my days into large blocks of time instead of interwoven meetings and seminars, because then I'll just get nothing done. But my life is also not sorted. I'm not used to having adjustments, so I don't really know what to ask for. I still get trapped in wishful thinking where I'll work well from home today and do nothing. I have many, many days where I don't get into the office before lunch, and even more where I don't even do more than a couple hours of work. I'm not even sure that my supervisors know what I struggle with, because I don't even think I know that myself. All I know is I'm trying my very best to just get through the days and deadlines as they come, figure out what works for me and what doesn't. And the one thing I do know, however, is that I really want to change things and spread awareness, to educate and inform those who may be unaware of what ADHD truly is, to bring some kindness and understanding into this world, to spare others the guilt and shame of living with undiagnosed ADHD and urge them to seek out a diagnosis if they relate to all or some of my experiences. Which is why, after having told only my parents and about 3 of my friends when I got diagnosed, in order of ADHD Awareness Month, I decided to announce my ADHD to my entire Instagram following and spend the entirety of October posting various infographics, reels, and memes about ADHD to my stories. I did get some mediocre responses because objectively it was quite a big spam, but I was really touched and positively surprised by the number of people who reached out to me, both over social media and real life, either relating their own experiences, commending my campaign, or seeking answers to their own situation. So what's next for me? Nothing new really, just trying to get through the PhD as best I can, continuously learn about myself and ADHD, possibly do a postdoc in ADHD, see about that, all while getting closer to figuring out how to fully accept myself, be open, raise awareness in the most meaningful ways possible, so that by the time I have children, and they likely, because it's strongly genetic, have ADHD, the world is a better place for them, and hopefully I'll get there. So yeah, sorry I went a little bit over time, but thanks for listening. Here's my social media if anyone's interested. I don't actually have the X logo, but I do have the Twitter logo. We all know that's the same thing. So that's it from my side. Thank you.
Thank you so much, Julia. It was such a wonderful worldview of ADHD. And of course, we can't really sort of glamorize it, but I can just say that from an audience point of view, it really hit the note, and we appreciate you sharing your journey so much. And if you guys have any questions, please leave it in the chat. We're going to address them at the end of the talk. Next, we have Sophia. Sophia is an MD PhD student at Albert Einstein College of Medicine in New York. Before moving to New York to pursue her graduate education, she received a bachelor's degree from UCLA in neuroscience, where she studied traumatic brain injury models in rats. Last year, Sophia was diagnosed with ADHD for the very first time at 23, and ever since she has been on her journey to figure out what the best strategies for her are to manage this condition, and also in her personal life as well as in the workspace. She has a fantastic Instagram page, which I personally am a big fan of. It's called Sophia in the Lab, and she uses that platform to share her difficulties of being a PhD student, including her experience with ADHD, in hopes of validating other people who are going through the same struggles. The stage is yours. We're very excited to have you.
Thank you so much, guys, for the introduction. And thank you, Julia, for all the background. I just want to say everything you said about yourself, I relate very, very much to. So that was great. So let's see. Oops. Can you guys see my screen?
Yeah,
yes, we can see it.
Okay, so I was thinking about how to organize this, and I decided to maybe not talk as much about the solutions that I came up with for managing my ADHD, but more about what kind of a little bit of an earlier stage of the ADHD journey, and I'll explain that in a second. So ADHD in academia, what not to do. So a little bit about myself first. So originally I'm from Russia, but I currently am in the United States. I got my neuroscience degree from UCLA, and then now I'm doing an MSTP or an MD-PhD program at Albert Einstein College of Medicine in New York. It's a picture of me being in a dissection room because I love surgery. Yeah, currently I'm in my PhD program years in the program. And just like Julia, I was diagnosed with ADHD last year at the age of 23 for the first time. And because it's, for me, it's pretty recent, even though, again, looking back, all the signs were there for years, but it's a pretty recent diagnosis for me. And so I don't think I have a good grasp on how to manage it well yet. It's a work in progress, but I think I kind of got to a stage of awareness and kind of maybe things that you shouldn't, the ways that you should not be looking at this condition. So I'm gonna talk about that a little bit. So one of the things I think happened to me, but I think it's not just specific to ADHD or any like neuro or like mental health diagnosis, is when you kind of obtain a new identity, when you become diagnosed with something, when you join a new like program, like being a grad student, you might often feel like you're not that enough. And that's something I've struggled with. So I felt after my diagnosis, I felt like, oh, I'm diagnosed with ADHD, but like I felt a little bit like an imposter in being a person with ADHD. I listed a few things that can make you feel like even if you receive some diagnosis, that actually that's not really real for you. Like it's a little bit like you're a little bit imposter in that field. So some of the things that if you get this diagnosis as an adult, so that if you're choosing to not use medication, I think that can increase that feeling. The fact that more and more people, especially around me in academia, I'm seeing being diagnosed with ADHD, and then also cultural background. So let me quickly cover the last 2 points, and I'm going to go more in depth with the first two. So a lot of my friends and colleagues, I've noted, or when we talk, it comes up, have been diagnosed with ADHD because now we're more aware of this condition, unlike 15 years ago. But to me, in the back of my head, I'm a little bit like, oh, is it just like now just more of us are getting like diagnosed, or is it, do I really have this condition, or is it just like I don't know, it's just more diagnosed and I don't really have it. That was like one of kind of like thoughts I had in the very beginning. And the second one, I came again, I'm from Russia, and in my family and in my culture, there's a lot of stigma around mental health. And so I think for my family, because even if I say to my parents, oh, mom, like I've been diagnosed with this, mom was like, no, this is just like, you're just so active. You're just not paying enough attention to this. Like I was like this, and if you just focus more, you can do this and this. So I think obviously like, I know myself that that's not correct, but it kind of like can get to your head a little bit and like doubt, like, are you like doubt your diagnosis. But the first two, I kind of want to talk about more in depth in regards to myself. So as I mentioned earlier, I was diagnosed last year, so I was 23. And again, as Julia said earlier, when you think about ADHD, just like for our first thoughts is like a kid in school who is like, I don't know, not raising his hand or getting the assignments late or getting bad grades. And I never really struggled in school. I always had also like very good grades as well as an undergrad. And then so I was thinking like, oh, if I manage my whole life, like like obviously I don't have ADHD. But then as I started thinking about more, it was like, okay, all of my assignments were like done like the night before or a few hours before the deadline. All my exams I studied for like the night before, and the only reason I got good grades is because actually I have like very good short-term memory. But actually I was not, and like my teachers all the time, they were telling me, oh, you're not paying enough attention, like pay more attention, you're doing very careless mistakes. So it all made sense, but because in my head I was thinking, oh, I managed my whole life to deal with this. Like, is it real, like that I'm being diagnosed now at 23? And the second point is that when I was diagnosed, the doctor asked me if I wanted to have medication for ADHD. And for personal reasons, for personal choices, I did not personally want to use any medications. And a lot of people, again, in my med school and then my grad school class, a lot of people, or I guess like all the people who I know who have ADHD, they are on medication. And so me thinking that I don't want to use the medication, I was almost feeling like felt very invalidating to myself, because I was thinking, oh, if I don't need medication, that means like you're not actually struggling. Like people who are struggling are using medications. And I think it took a little bit of grace onto me to say that it's even if I'm not deciding to not use the medication, deciding to work through it or figure out different ways, that doesn't make it less real, like what I have. I'm not a fraud who's pretending to have ADHD to cover my own mess. Like it's a real condition that I have, and I'm just choosing a slightly different path. It's not less valid. So kind of like the takeaway from this section is that even if you see a lot of people around you, if you get some diagnosis and you decide to take a path that's a little bit different from the majority of people you see around you, that doesn't mean your like condition is not real. That doesn't mean you're just lazy or messy. It doesn't mean you're doing something wrong. You can try to explore it on your own path. Like it's okay. And the second kind of takeaway I have, I think it flows a little bit from the first one, in the sense that when you think, especially that maybe your condition is not like, it's not real, or it's not as real as other people's, you oftentimes you attribute the symptoms of your condition, or for me personally, I oftentimes do that. I attribute these various symptoms to just my personal flaws, and then I blame myself for them. And then I think, oh my gosh, I'm such a horrible scientist, I'm such a horrible person. And I wanted to like list a few things that pertain especially to me working in the lab, things that I struggle with. For example, because I have like pretty bad time blindness, it's very hard for me to estimate how long what task is going to take. Sometimes like I say, oh yeah, guys, I'll be done. Like if my friends are asking me for coffee, I'm like, yeah, I'll be done in 30 minutes. And then an hour later, I'm still like very much in the middle of what I'm doing. And because of that, it's very hard for me to plan like my day, because I don't know how much time which task will take. And then my grandmother, who is not a strong believer in mental health and like mental wellness, she's always like, oh, just plan your day and then just do whatever's on the plan. And it's really hard to do that. Yes, like having a plan can help, but it's really hard if like you can't estimate how much time it's going to take. Another thing for lab, which is really important, is when you do your experiments, is that you actually like, if you have your Western blots, you scan them and put them like in a presentation. You curate figures, and like you take them from Prism or whichever software you use them, and actually save them in the same folder for your experiments. And it's once I'm actually doing the experiment, I have the motivation to finish it. But once I'm done with the experiment, my motivation level for this specific like experiment like falls very, very low. And then it makes it difficult for me to care about like the actual like the data that I got, and like to put like keep it organized. And it's really important to have the data organized, but it's really hard to do for me. It's really hard when I have like a few different experiments to not get overwhelmed in my head, thinking like I'm doing one experiment, and at the same time in my head I'm like, oh my gosh, this other experiment. So that can be really hard, really forgetful, which is a little bit embarrassing, because I would ask my lab member, I was like, oh, what's the concentration you said if we were like working together? I'm like, oh, what's the concentration? And she's like, I don't know, like 10 micrograms or something. per microliter. I don't know, I'm just making this up. But she would say the number, and then I'd be like, okay, got it. I would go, like, I don't know, like drink some water, like think about something else, and like, oh shit, like I completely forgot what she said. And then I have to go back and ask it again. And then difficulty focusing during the day, and as well as like, this is just a funny one, I keep leaving my like pipettes or like tube racks or like ice buckets like everywhere, and then I keep like walking around the lab and trying to find them. So all of these are these are symptoms of my condition that are appearing in my lab work, but oftentimes as they're happening, I kind of forget about the fact that it's not that I'm lazy, I don't want to focus, I don't want to like plan my day. It's like, no, I really do want to plan my day, but it just doesn't always work out the way I expected. And then when it doesn't work out, I feel really bad and like, oh my gosh, like, I did such a horrible job. Like I suck at this. And then for other things, I'm like, I don't know, for a lot of these things, oftentimes I forget about my condition, and then I just end up blaming myself for these like mistakes that I do. And I think it's really important to give yourself grace and kind of try to remember that it's not your fault, like you don't suck. You're trying your best, you're not just lazy. And I think especially I think it's hard when you're comparing yourself, when you're working in a lab or in a group of people, and then maybe, of course, we don't know who might have what kind of struggles, but if there are other people, for example, who are maybe struggling with ADHD less or don't have ADHD, and you're comparing just based on performance, that's not really fair to yourself. So I think the second takeaway is like to try not to compare yourself to others, and kind of just you're on your own journey. You are doing your best, even to yourself it might not look like that all the time. But don't attribute or condition. Don't make it seem like it's your own character flaw, and then punish yourself for that. Yeah. And in terms of ways to cope with different things, I'm still trying to figure it out, but I thought that I can share with you guys these kind of don'ts of ADHD.
Thank you, Sophia, for the talk and the realistic ideas surrounding ADHD, especially the bits with imposter syndrome really hit home. We would like to thank all our speakers for their time and valuable insights. It was an absolute learning experience for all of us here. So what's next for WIN UK? We run events in themes. We run WIN UK Research, WIN UK Reality, and WIN UK Network every month. If you have more ideas or have any questions, please do drop us an email, which is just on the slide.
So we're also very excited to present our next event, which is going to be an applications Q&A, and it's going to take place also virtually on MS Teams on the 13th of December. It's the most comprehensive one-stop shop for the application advice that you might need. We're going to be joined by postgrads, postdocs who are going to share their experiences of what works for them, and you can get good insights from that for your own application process, I feel. More details will be supplied on the social media and website stuff, so stay tuned for that. Next, we'd also love if you guys would stay in touch with us. You can sign up as a member on the WIN UK website. You can also comment on blogs and chat on our various social media platforms that we have. You can also feel free to email us. We have a Women in Neuroscience UK general email, which is administered by Lizzie, but we also have a specific one for events, blog, and the secretary team. With that, I'm going to hand it over to Shloka for the feedback form.
Don't forget to fill in our feedback forms. That is important because that just lets us know how we've done and what more you'd like from us.